Hello, welcome to this virtual poster hall. And in this poster presentation, I'm going to talk about our research identifying genomic cis-regulatory module interactions in Drosophila using deep learning. And this is the presenter, Tzu-Hsien Yang, and I'm from National Cheng Kung University, Taiwan. In this study, we try to study transcription regulation. As we all know that cells can respond to external stimuli by regulating the transcription of a certain set of genes. Transcription regulation in metazoan species, especially animals, is controlled in a modular manner. This means that different sets of transcription factor binding sites, or the DNA sequences where the transcription factor binds, can cluster together, and this clustering of transcription factor binding sites can cooperatively recruit the TF's transcription factors that have interactions. And through these transcription factor interactions, they can control the expression of their target genes. And the clustering of these transcription factor binding sites are called cis-regulatory modules in this research, or CIMs. Recently, it is shown that CIM themselves can interact with one another. For example, the enhancer and promoter CIMs can interact with each other to regulate their target genes. And these CIM interactions are usually formed by 3D chromatin configurations. And researchers have already shown that there are substantial complex diseases influenced by CRM interactions. And this provides another regulation layer in metazoan species transcription regulation. In order to study the interactions among CRMs, we can resort to experimental approaches. For example, we can use chromosome conformation capture sequencing technologies or the Hi-C techniques to probe the interactions among DNA regions. But if we want to apply Hi-seq to identify CRM interactions, it will require very large Hi-seq reads, which means that it will cost a lot of money if we want to perform such experiments. So a better idea is try to use some computational tools to prescreen the possible candidates. But existing tools considers only enhancer promoter interactions. They ignore other types of CRM interactions. For example, the enhancer-enhancer interactions or insulator-insulator interactions are all ignored by existing tools. So the goal of this research is trying to develop a general CRM interaction identifying tool, and we will use deep learning to achieve this goal. Before we can develop the deep learning model for CRM interaction identification, we first need to prepare the ground truth dataset. In this research, we prepare the ground truth dataset by analyzing the Hi-C experimental data. And we analyze the Hi-C experimental data by mapping them back to the Drosophila reference genome and use pair tools to find the interacting pairing sequences. We also collected verified CRMs in order to generate CRM interaction pairs. And based on the Hi-C interacting pairs identified by pair tools, we try to overlap these pairs with those verified CRMs. On the left-hand side, we try to generate the positive interacting CRM pairs. And we first filter out those confident Hi-C pairs. We ensure that these pairs are of only UU, UR, and RU types. And we also require these Hi-C pairs to have sequence reads more than 10. And we overlap these confidence Hi-C pairs with the verified CRMs. Then we get these CRM pairs. And in order to get the confidence CRM interacting pairs, we further require the CRM pairs to have more than 10 confidence Hi-C pairs. We also retain only the non-overlapping CRM interacting pairs, and in total, we get 27,507 positive interacting CRM pairs. On the right-hand side, we try to generate the negative non-interacting CRM pairs, and we first define the loose Hi-C pair And the criterion for loose Hi-C pair is they are of UU, UR, and RU types, and the reads of them are at least 1. And by overlapping the loose Hi-C pairs with the verified CRMs, we can get the loose CRM pairs. And we then sample CRM pairs that do not belong to the loose CRM pairs so that we can get those non-interacting CRM pairs from the Hi-C experiments. And the final positive and negative data are balanced. They have the same number. In this slide, we show the brief sketch of the constructed deep learning model for identifying CRM interactions. And this model can be divided into 3 different parts. In the first part, the model will accept 2 CRM regions as its input. And for these 2 CRMs, the model will first extract the feature experimental data from 4 different types of data. The first one is histone modification data. The second one is chromatin binding protein target site data. And the third one is the nucleosome-free site data. And the last one is the data of TF transcription factor binding site patterns. Based on the extracted experimental data for these 2 CRMs, the model then utilizes a consecutive 1D convolution operations to help obtain the most significant combinations of these features. Then in the second part of the model, we utilize the channel attention operation to help calculate the importance of different feature combinations. And then the cell attention operation is used to merge the information of these 2 CRMs. Finally, in the third part of the model, this information are all classified based on fully connected layers. And the output will be interacting pair or non-interacting pair identification for these 2 given CRMs. This is the model performance of the constructed deep learning model for identifying the CRM interactions. As we can see from the RC curve and the PRC curves, the test performance of the constructed model achieves more than 90% of area under the curve regions. And for the F1 values, we achieved about around 90%. So from all these performance metrics, we can see that the constructed deep learning model can indeed identify the CRM interactions in cells. That's all for the poster presentation. Thank you for listening. And if you have any questions, you can contact us and we can have a small discussion on the topics presented here. And thank you again.